Is Ben Walsh. He is working with Jonathan Simon, right? Professor Dr. Jonathan Simon. He is a master's student. You did your undergrad, did his undergrad here as well. And he's a Northrop Grumman fellow, is that correct? Yes.
Yeah, I guess so.
All right. And yeah, so take it away.
Okay, thanks for having me. Today I'll be talking about analysis of gamma band auditory responses in schizophrenia. So everyone here has probably heard of schizophrenia before. You may not know it actually affects about 1% of the general population. That kind of struck me as a larger statistic than I expected. And the standard diagnosis is based off of this DSM, or Diagnostic and Statistical Manual of Mental Disorders, which gets updated from time to time. It's on the 4th edition right now, but basically it's this clinical interview. It's an inherently subjective measure, and that's the standard diagnosis. The manual lists a lot of symptoms that you may associate. In particular, I think there's a perception of hallucinations being a common symptom, which is true. And auditory hallucinations in particular, what you might describe as voices, are experienced by almost all schizophrenic patients at some point in their life. And because of these auditory deficits, there have been experiments for decades, basically, researching, trying to quantify what are the differences in auditory responses. And in particular, gamma band, so between 30 and 50 hertz, this band has been a good candidate, and it's been known for decades basically that a lower response, in particular a steady state response, is found and is significant in schizophrenia. So the basic experiment here, or the data, is from 50 schizophrenic patients who are age and sex matched with 50 controls, about middle age, no one too young, too old. Sometimes these types of experiments are with amplitude modulated stimuli. In this case, they're clicks. And if you haven't seen a click, it's basically square waves, and which is actually very broad in frequency. And it's presented at 40 hertz. So even though it's 15 clicks, it's actually very, very quick. And so it's less than half a second is one trial. So you're presented with 75 trials, and then the gap between trials is varied a little bit, so you can't anticipate when the click is.
What is the y-axis?
Oh, well, this is literally just the wave data in this case. So this is like literally the sound file. So there's no, yeah. Or I guess it'd be sound pressure, and then this is just the amplitude of the frequency response, just showing that it's broadband, even though it's presented at 40 hertz. Does that answer your question? Or?
So what are those nulls coming from? In frequency?
I mean. So I guess when the clicks come, I mean, so these are actually this is cut off actually. This is, I think, times 10 to the something. But in a click, you basically get harmonics at 40 hertz. So all I was trying to show here, I mean, I didn't choose this stimulus, but all I was trying to show is that when you have these clicks, even though it's basically these train of square waves, you get very broad frequency.
Function, right? The Fourier transform of the square waves.
Yes. Yeah, exactly. Yeah.
Oh, okay.
Yeah. And so this data is coming from a collaboration with Elliot Hong from the Maryland Psychiatric Research Center, from the Maryland Medical School, and this is from a seed grant between Dr. Simon and Dr. Hong. Okay, so that's what subjects are presented with, that's what the stimulus is. To record the responses, we're using EEG, electroencephalography. And if you're not familiar, in general, EEG is measuring these potentials generated by large populations of neural currents in the brain. So about 10 to the 4 neurons or so. It's a complementary modality to MEG or magnetoencephalography, which is used heavily in our lab in general. And EEG in general it's a good modality for auditory experiments because it has high temporal resolution, it has moderate spatial resolution, and it's also non-invasive and it's quiet, so it's good for auditory experiments. And so that's EEG in general. In this particular case, there's 64 electrodes from this data set. The sampling rate is 1 kilohertz, which is, so getting about that millisecond time resolution. You keep the impedance at a certain value. And if you've never seen a EEG cap before, that's one version of what it can look like. So you just sort of strap it on, and it's not uncomfortable or anything. Okay, so for processing the data, it's bandpassed, and this is sort of a general frequency range, but in general you want to cut off the first couple hertz for biological noise of no interest, and again with a very, very high frequency noise such as power. Unlike MEG, for EEG, since you're measuring a potential, you need some sort of reference, and you need some sort of ground. So in this case, we sort of take a virtual ground by re-referencing over the average of the channels that are not abnormally high in power. Another thing we do is we detrend it. We do this linearly. We do it over time, and after we split into trials, which I guess I didn't specifically say we do here, but the voltage can drift over time of the electrodes. And a standard algorithm we use in the lab for denoising environmental noise in particular is time-shifted PCA, or principal component analysis. And as part of these of the electrodes in the cap, you have ocular channels. You have channels that or electrodes that pick up responses that are not associated with auditory responses, and so these form a basis for artifacts that we can remove. And as sort of a simple dimensional reduction, we actually only take, in this particular analysis, a single electrode. And so this may seem strange, but it's actually not uncommon to take just a single electrode. And it's good to keep in mind that through the rest of the pre-processing, information between the other electrodes is sort of incorporated into what's in this CZ. And to give you at least a little bit of motivation for why this might not be an oversimplification. Sort of a typical auditory head map is shown there on the top right. And you can see if you were just going to pick the electrode with the highest variance, that actually would be CZ. And so that varies a bit. Maybe you take the electrode right in front of it, but it's actually a fairly common dimensional reduction technique. Okay, so the workhorse of the analysis here is going to be a Gabor filter. Yeah.
Why are you doing any analysis on the signal in the first place? You want to do what, like distinguish between like people who have this illness or
Yeah. Yeah, as I said before, it's been known there are certain deficits in auditory processing and schizophrenia. And so the question is, can we characterize them more? And can we, as I said, the standard assessment is, the standard diagnosis is subjective. So can we get a more objective measure? And not just be able to say what quantitatively is different, but maybe give some sort of explanation of the underlying neurobiology, why this even happens in the first place. So for a Gabor filter has been used to illustrate responses to different modulation frequencies in EEG and MEG, and it gives a good balance between frequency resolution and time resolution. So you can see how the response at a particular frequency evolves smoothly over time. And basically all it is just a Gaussian times a complex exponential. And so sort of what you might expect, here's the real part of the filter because it's complex. You kind of have this envelope of a Gaussian, but then you also have this frequency. So that's a general Gabor filter. And from the outputs of this filter, you have a real and imaginary part. And so you can take a look at the energy, which is phase insensitive. And to give you a sense of what the energy might look like, so this is from one of the previous papers I just mentioned that use this. This is for MEG, but to give you a sense, if the filtered signal, has some sort of onset component, and then it has sort of a steady state part, and sort of visually you can see there's this frequency, then you get this envelope, which is at that frequency. And then you can also look at the phase, and this is just sort of a fancy way of doing the 4-quadrant phase component. So from the phase, we wanted to look at the phase coherence. And so just introducing some notation of the ith trial and the nth subject. If you look at the phasor data, at unit phasors, typically the coherence would refer to the inter-trial phase coherence. So as I mentioned, there are 75 trials, and from trial to trial, you have a phase over time. And so you can imagine if you sum all the phasors from trial to trial, and they all had the same phase, and then you took the magnitude, then if they all align, then this coherence would be 1, and if they're all random and canceled each other out, you'd take the magnitude and it'd be 0. So that phase coherence is going to be a metric of how consistent the response is from trial to trial, and it's going to vary from 0 to 1. That's sort of the typical metric for coherence. Yes, Andrew?
So but phase coherence, is it the same thing as time delay? So you input a stimulus and then you look at when the biological system outputs a response to the stimulus?
So not necessarily. So this phase coherence is going to be a function of time. So, depending on what the delay would be, there's going to be some associated phase. But if that delay, as long as that delay was consistent from trial to trial, you would have a high coherence. So intertrial phase coherence, I'll just call it ITC because I'm going to be saying it a lot, is sort of the typical metric of coherence. And if you just sort of switch around these variables, you can also look at the ISC, which I actually haven't seen reported, but the same idea. Each subject has a certain phase, and if all the subjects had about the same phase, then the coherence would be close to 1. If they all had just their own phase, then the coherence would be 0. So first, just to give you a sense of what this data would actually look like after the filtering and the dimensional reduction, you see that the frequency is present in kind of the first stage. It's sort of ramping up a little bit. And I don't know if you can see, but I tried to just show very lightly, but probably too lightly, the individual trial data. I don't know how well you can see it, but if you see this sort of vague cloud of light blue, what I was trying to illustrate is that there is variation from trial to trial.
So each of those peaks correspond somewhat with a click that they're hearing?
Not necessarily. So I mean, I guess what I could show is on the same graph is when the clicks were responding. So I mean, basically every click is going to elicit this pulse, but it's not that there's a click here, there's a click there, there's a click. As Andrew said, there's this delay, which varies, but there is a delay.
Is this coherent or not?
Well, I will get to that. I just sort of wanted to show what it.
By looking at it, can you say something?
Since you averaged over these trials and you did get a frequency, it is coherent at some level. You could say that. So first, looking at the energy at 40 hertz, this is sort of what we expected. During the stimulus presentation, you see the energy at 40 hertz. It's smooth because of this filter, and it ramps up over time. And in general, a healthy control is going to have a stronger response than a patient. The main areas of difference would be right around the onset and sort of in the middle of the stimulus presentation. And so this is interesting to see over time where in particular the differences are. A graph like this in another paper might just be summarized by the magnitude of the FFT at 40 hertz. So now actually looking at exactly what the coherence is and how it changes over time. So again, blue are the healthy controls and red are the patients. So we sort of expected this that around the onset and just in general, the healthy controls would have more coherence from trial to trial. It was actually more similar than we would have expected, but again, sort of around this onset area and kind of in the peak during the stimulus presentation, the ITC was higher for controls.
Excuse me, can we get back to the slide and define this?
Oh, sure. Right, so the data is, if you convert them to unit phasors and you sum them up, if all the phasors align and then you take the magnitude, then the coherence would be 1. If they canceled each other out, then the coherence would be 0. In practice, if you have 75 trials, you're definitely going to be somewhere in between, but that's a basic idea.
Also, all this analysis is looking at 40 hertz, right? All that your Gabor filter is 40 hertz.
Right.
And that's because your input stimulus is 40 hertz.
Correct.
But did you consider doing something like a spectrogram and you look at a whole range of frequencies to see if stuff, because I would imagine you can generate additional frequencies.
Right, so I did not filter this data at different frequencies. The data set was part of a very large comprehensive data set which had 20 hertz, which had 10 hertz, which had 5 hertz, so I did look into that, and basically around 40 hertz was sort of the most promising results, which is not unexpected, just given historically people have always looked at 40 hertz because there's a big difference. Yeah. So to kind of recap, the power at 40 hertz is stronger in general over time in controls, and the ITC is higher in controls. And now for the ISC, it's like the graph just switched. This is not something we expected. This was unexpected that the patients actually around this onset area, it doesn't exactly overlap, but there is a higher ISC, in particular around the onset area, and it's higher in general around in the middle of the stimulus presentation. So this was something that was unexpected. The first time it came about, I actually thought it might have just been an error, because it does sort of look like the graph just switched. But this is what we're seeing, that although the healthy controls have a stronger response, and from trial to trial the response is more consistent, if you look at how the phase varies across a group, the healthy controls have a more random phase, their phases are more different, while the patients have a phase which is more aligned. So, how is this possible? Well, we started thinking about sort of a general model that could explain this. So, starting off with sort of the general part of it, that there's some basically 40 hertz component with some associated phase, and there's all sorts of other noise. I'll just call this N of T. It incorporates a lot of parts. You observe the signal with the noise, and this phase here, part of the 40 hertz response, is going to have some variance, which is part of the ith trial and for an nth subject. So to model these differences, so for the ith trial, we just said, okay, well, the ith trial is going to vary as a normal variable, and just in general, we're going to say that the variance for a patient is higher. So they're both going to be random, but the component that changes within the trial is going to have a larger variance for the patients. And here, just with the constants associated with the signal and with the noise, we're just going to say that there's a higher SNR for the controls. The responses, there's a higher SNR. And now for the phase, for the nth subject, sort of the base phase, we're modeling this as, oh, I actually switched these, sorry. The controls just have a random phase, while the patients have a randomness but centered. And I need to make a note of that. Okay. So this model actually works, and right now it's not tuned to exactly replicate the numbers of what the ITC and ICC were, but in general, if this model outputs a higher energy from the Gabor filter at 40 hertz, it has a higher ISC for the patients, and it has a higher ITC for the controls. So that's one, it's a possible explanation, but then why would this be the underlying model in the first place? That controls would have a higher response, be more consistent from trial to trial, but their individual phase would be more varied. Well, a similar effect has actually been seen in other EEG experiments, in auditory experiments, and EEG, in visual experiments, excuse me. And so in Henry et al., for instance, they were seeing that they had a metric for gap detection, so performance on the y-axis, and they were FM modulated, but FM stimulus on the x-axis, and they showed just a few individual participants' data. Well, these curves are basically modulated at the same frequency as the FM modulation, but you can see from subject to subject that the different phase of these different curves is different. So it's as if individually these a healthy control has a sort of phase optimization internally. That the phase is definitely important, but there is a different preferred phase from one person to the other. So overall, the reduced power at 40 hertz and reduced ITC at 40 hertz in schizophrenia, we found these. This was sort of a sanity check. This was consistent with previous literature. But the increased ISC, or the coherence over subjects, that we found, this was unexpected. But we are proposing this phenomenological model, which is consistent with the experimental observations, and moreover, it does align with other effects seen in other experiments, and by suggesting this phase optimization or lack of internal phase optimization or finding a preferred phase in auditory processing at this gamma band at around 40 hertz, it's offering an explanation for the auditory deficits that are seen in schizophrenia. And I'd like to thank my advisor, Dr. Simon and Elliot Hong, for collaborating here with the seed grant, and the primary lab members, Francisco and Krishna, and funding, and thank you for having me.